Skeletal muscle alanine is synthesized in the muscle from products of both protein breakdown and glycolysis. In the human body, skeletal muscle is the primary site for the endogenous synthesis of alanine, meaning it manufactures this amino acid from scratch. The synthesis requires two key raw materials from within the muscle cell. First, the nitrogen source. This comes from the breakdown of muscle protein, which releases branched-chain amino acids, or BCAAs, valine, leucine, and isoleucine. These BCAAs act as the donors of amino nitrogen. Second, the carbon source. This comes from glucose. When muscle breaks down glucose for energy via glycolysis, it produces pyruvate. The actual assembly of alanine is catalyzed by the enzyme alanine aminotransferase, or ALT. This enzyme transfers the nitrogen group from the BCAA-derived glutamate to the pyruvate molecule, resulting in the formation of new alanine. This newly synthesized alanine is then released from the muscle into the bloodstream through specific transporters to be sent to the liver. Next, we will move into alanine released and transport from muscle to liver. In this section, I will explain what happens to alanine after it is produced in skeletal muscle. After being produced in skeletal muscle, alanine is released into the bloodstream. Alanine then travels through the bloodstream to reach the liver. The liver takes up alanine for further metabolic processes. In hepatocytes, alanine is transaminated by alanine aminotransferase to form pyruvate. Importantly, alanine acts as a safe nitrogen carrier, transporting ammonia from skeletal muscle to the liver without causing toxicity. Once alanine reaches the liver, it enters the glucose-alanine cycle, which will be explained in the next section.
Next, we will proceed with the liver function in glucose-alanine cycle. This is the overview of glucose-alanine cycle. Skeletal muscle produces alanine, which travels to the liver. In the liver, alanine is converted into pyruvate, which is then converted into glucose. At the same time, the nitrogen is removed as urea and excreted by the kidneys. Let's look at each step in more detail. The skeletal muscle produces alanine from pyruvate and nitrogen released during protein breakdown. Alanine is released into the bloodstream, carrying nitrogen safely to the liver. This allows the muscle to remove waste nitrogen while supporting energy production. Once alanine reaches the liver, it is converted into pyruvate. During this process, the nitrogen is removed from alanine, which will later be processed into urea for safe excretion. The pyruvate generated is then used in gluconeogenesis to produce glucose. The glucose formed in the liver is released into the bloodstream and transported back to the muscle, providing an important energy source during fasting or exercise. Meanwhile, the nitrogen removed from alanine is converted into urea in the liver. Urea travels through the blood to the kidneys, where it is excreted in the urine, safely removing waste nitrogen from the body.
Why does the glucose-alanine cycle exist? The glucose-alanine cycle plays a dual protective and metabolic role in the body. First, it acts as a nitrogen disposal mechanism. Second, it supports energy balance, especially during times of increased demand. The cycle removes toxic nitrogen from muscle while supplying glucose back to it. When is the glucose-alanine cycle activated? The glucose-alanine cycle becomes more active during catabolic or stress states, including fasting or starvation, prolonged or intense exercise, illness, trauma, or infection, poor carbohydrate availability. Next is hormonal regulation. The cycle is regulated mainly by catabolic hormones, cortisol and glucagon. Cortisol increases muscle protein breakdown and provides amino groups for alanine formation. Meanwhile, glucagon stimulates gluconeogenesis in the liver and enhances conversion of alanine into glucose. At the same time, insulin inhibits this cycle by reducing muscle proteolysis and also promoting glucose uptake and storage instead of glucose production. Next is the importance of glucose-alanine cycle. It prevents ammonia toxicity by converting muscle amino groups into non-toxic alanine, shuttling them to the liver for urea cycle processing instead of dangerous free ammonia buildup. This protects brain function and systemic health under protein breakdown stress. Metabolic recycling. Alanine supplies pyruvate for liver gluconeogenesis, producing glucose that returns to muscles for ATP via glycolysis, sparing muscle protein longer than lactate-based cycles. Endurance efficiency. By optimizing NADH use and carbon export, it boosts muscle energy yield during prolonged activity, linking catabolism to anabolism for survival advantage.